All right, guys, I'm gonna attempt my first screencast to help you out with a few sections of this PowerPoint. Hopefully this will work. So the first few slides I think you guys will get, it's a little review of Mendel and meiosis. And the first day's activities kind of laid the ground for the basic stuff, simple inheritance patterns, basic Punnett squares, and today it gets a little trickier. So here we go. Hopefully you can see this screen titled Non-Mendelian Genetics. The first one I want to explain is incomplete dominance. And just like the heading says, this is a type of inheritance that is basically like a blending of the traits. So you can see the parental generation is you can see the parental generation here is a purebred white parent crossed with a purebred red parent. Notice that the alleles are homozygous recessive crossed with a homozygous dominant. So if this were normal Mendelian genetics, we would have an F1 generation of 100% red heterozygotes, but you guys can see that's obviously not the case. Instead, we got pink. So 100% of our F1 generation are heterozygotes still, but they are pink and not red. So this is an example of non-Mendelian genetics where the traits are blended. Think of it like mixing of paint. Now, if we do another cross and do an F2 generation, you can see that then we get back to kind of our standard ratio. Well, not standard ratio, but we can see where all the colors and all the phenotypes, that's a new word for you guys, the physical traits, the phenotypes, in this case flower color, are visible. So we see our homozygous dominant. We have 1/4 of a chance of getting red. We have 50% chance of getting heterozygous, which is our incomplete dominant pink, and then a 25% chance of getting our homozygous recessive white. So as you can see, this is just different from normal inheritance, which is why we call it non-Mendelian genetics. He was not aware of how all this worked, so he didn't see this stuff. Okay, let's go on. So for our next section, this one is called Codominance, and apparently I have to delete these little marks. That's going to be irritating. This is called Codominance, also another example of non-Mendelian genetics. And as you guys can see, we're kind of looking at a similar example. We're looking at flower color again, and this time we're crossing a red camellia with a white one, but instead of getting pink as we would with incomplete dominance as our phenotype, you can see that both alleles are equally expressed. So if you look at the flower below, in the example, you can see that we have a uno momento. You can see here, nope, that didn't work. Anyways, you can see in the picture that there is a red and white flower. So instead of being pink, we have red and white, which is our example for flower colors where we can see both alleles are expressed. Okay, let's go to the next one. Sorry, figuring this out. I promise I'll get better. So with this is just showing you guys how you might do a Punnett square for like this codominance example. So you can see that we kind of use different looking alleles just to sort of emphasize that we're looking at non-Mendelian genetics. So our parent is you can see right here, our parental generation, we have a purebred red crossed with a purebred white. So think of, you know, the gene as color for C and the allele as R for red or W for white. So when you do this cross, you'll see that just like in the codominance cross, we don't get our normal predicted inheritance pattern. Instead, we get 0 red, 4 red and white flowers, and then 0 white. So the heterozygotes, one more time, are codominant. They show both alleles equally. So if we're using our paint analogy again, this is like a splattering of paint for codominance as opposed to a blending of paint like it was for incomplete dominance. We are going to keep going over this, but this is basically just more examples of Punnett squares that you guys can flip to, and it shows you various examples for different types of crosses. Single factor Punnett squares like these are not too bad. I'll probably have to do a video when we do our 2-factor crosses. But click through these and there's lots of tutorials online on how to do single factor crosses. So I'm going to click through, keep going. Test crosses. So test crosses are a little confusing. Basically, it's a way to determine the genotype of an unknown individual. So like often when you look at a flower, I say, hey, that's a red flower, that's cool. But since red in this case is dominant, we don't know whether this red flower is homozygous dominant or a heterozygote. So the way that you figure that out is you always cross your unknown with a homozygous recessive. The homozygous recessive, we know for sure, like if we know that red is dominant and white is recessive, then when we cross these two, we can see the different results. So for example, if we did a test cross and we get 100% red, that tells us that most likely the phenotype of the unknown parent is homozygous dominant. And then it's obvious if we get even one white plant that the unknown parent had to be heterozygous. So that's essentially what a test cross is, and that can be handy for down the road. Okay, I'm going to pause here and see how this video worked. Blood typing is no joke, so that will require a brand new video. Hopefully this works for you guys, and if this is super annoying, just go through the PowerPoint as is. I know I hate hearing my own voice on video, so hopefully you guys won't hate it as much as I do, and hopefully this helps. Best of luck. Shoot me questions through Canvas discussions. Thanks, guys.